Step 1. Understand what an eyepiece graticule is. It's a glass disc with a scale marked on it. It's placed in the eyepiece of a microscope. It appears superimposed on the image of the specimen. Step 2. Analyze what calibration means. Calibration determines how many actual micrometers, μm, correspond to each division on the graticule. This is done by comparing with a stage micrometer of known measurements. Different objective lenses will give different calibration values. Step 3. Evaluate each option. A. An eyepiece graticule can be used to make measurements. This is correct. After calibration, you know what real distance each division represents. This allows actual measurements of specimens. B. An eyepiece graticule is magnified by the objective lens. This is incorrect. The graticule is in the eyepiece, above the objective lens. It's not magnified by the objective. C. An eyepiece graticule magnifies the specimen. This is incorrect. The graticule doesn't magnify anything. It's just a measuring scale. D. An eyepiece graticule makes comparisons. This is partially correct, but not the primary reason. While you can compare sizes, the main purpose is measurement. Final answer. A. An eyepiece graticule can be used to make measurements. Explanation. The fundamental purpose of calibrating an eyepiece graticule is to enable accurate measurements of specimens. Without calibration, the divisions on the graticule would be meaningless. Calibration establishes the relationship between graticule divisions and actual distances, making precise measurements possible. One. Key features visible in the image. Large central nucleus. Membrane-bound organelles. No visible rigid cell wall. No visible chloroplasts. Various small vesicles/organelles in the cytoplasm. Cell membrane appears flexible. 2. Analyze each option. It is an animal cell because it does not have a cell wall. Correct. The image clearly shows no thick cell wall structure. The outer boundary is flexible, characteristic of an animal cell membrane. B. It is an animal cell because it contains a permanent vacuole. Incorrect. While animal cells can have vacuoles, permanent large vacuoles are characteristic of plant cells. The image doesn't show a prominent permanent vacuole. C. It is a plant cell because it contains many chloroplasts. Incorrect. No chloroplasts are visible in this image. Chloroplasts would appear as large, dark, oval-shaped structures. D. It is a plant cell because it contains many lysosomes. Incorrect. While lysosomes may be present, they are found in both plant and animal cells. The presence of lysosomes alone cannot determine cell type. The correct answer is A. It is an animal cell because it does not have a cell wall. The key distinguishing feature is the absence of a cell wall, which is a defining characteristic of animal cells. Plant cells always have a rigid cell wall surrounding their cell membrane, which is not visible in this micrograph. Looking at the Venn diagram, the left circle represents features related to messenger RNA and ADS ribosomes. The top circle represents ability to respire. The right circle represents containing circular DNA. The regions B and D represent overlapping features. Key to interpretation. 1. Region B is the intersection of all three circles. 2. Region D is the intersection between messenger RNA/ribosomes and circular DNA. Since we're asked about features found in both eukaryotes and bacteria, we need to check which of these labeled regions best matches this criteria. Analysis. Eukaryotes typically have 80S ribosomes, while bacteria have 70S ribosomes. Both eukaryotes and bacteria can respire. Bacteria have circular DNA, while eukaryotes typically have linear DNA. Both eukaryotes and bacteria use messenger RNA, though the processing differs. Looking at the diagram's regions. Region A equals respiration plus mRNA features. Region B equals intersection of all features. Region C equals respiration plus circular DNA. Region D equals mRNA features plus circular DNA. The correct region is region C, as it shows features that are definitively shared between eukaryotes and bacteria. Both can respire, metabolize. This is the only region that represents truly shared features without including features that are different between the two types, like ribosome size. Therefore, region C represents the features found in both typical eukaryotes and typical bacteria. 1. First, let's identify structures bound by a single membrane. Endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, vesicles. 2. Now let's analyze each cell type. A. Ciliated epithelial cell. Has many mitochondria, double membrane. Contains ER and Golgi, single membrane. Many vesicles. Specialized for movement with cilia. Moderate amount of single membrane structures. B. Goblet cell. Specialized for secretion. Has extensive ER and Golgi apparatus. Many secretory vesicles. Very high proportion of single membrane structures. Most of cell volume is taken up by secretory apparatus. C. Red blood cell. No nucleus. No organelles in mature form. Almost no membrane-bound structures. Filled mainly with hemoglobin. Very low proportion of membrane structures. D. Companion cell. Part of phloem tissue. Has normal complement of organelles. Some ER and Golgi. Moderate amount of single membrane structures. The correct answer is B. Goblet cell. Reasoning. Goblet cells are specialized secretory cells. They have highly developed ER and Golgi apparatus, both single membrane. Contains numerous secretory vesicles, single membrane. Most of their cellular volume is devoted to these secretory structures. Their primary function of protein secretion requires extensive membrane-bound organelles. Other cell types either have fewer single membrane structures or have more of their volume taken up by other components. 1. Understanding phospholipid structure. Phospholipids have a phosphate head and fatty acid tails. The head contains a phosphate group, PO4 superscript 3. 2. Analyzing each option. A. The phosphate heads are joined to water molecules by hydrogen bonds. While phosphate heads can form hydrogen bonds with water, this is a result of polarity, not the cause. This is incorrect. B. The phosphate heads are insoluble in water. This is incorrect. Phosphate heads are actually hydrophilic, water-loving. Insolubility would indicate nonpolarity. C. The phosphate heads become ionized in water. This is correct. Phosphate groups, PO4 superscript 3, become ionized in water. Ionization creates a negative charge. This separation of charge creates polarity. This is the fundamental cause of the polar nature. D. The phosphate heads are joined to water molecules by covalent bonds. Covalent bonds are permanent bonds. Water molecules interact with phosphate heads through temporary interactions. This is incorrect. The correct answer is C. The phosphate heads become ionized in water. Reasoning. Ionization is the process where molecules gain or lose electrons. When phosphate groups ionize in water, they develop a negative charge. This separation of charge creates polarity. This is the primary cause of the polar nature of phospholipid heads. The other options describe either results of polarity or incorrect interactions. 1. Statement 1. 3 cellulose molecules coil around each other to form a triple helix structure. This is incorrect. Cellulose molecules form parallel chains. They do not form triple helices. This is more characteristic of collagen. Cellulose forms straight, unbranched chains due to beta-1 for glycosidic bonds. 2. Statement 2. Many hydrogen bonds form between adjacent cellulose molecules. This is correct. Multiple hydrogen bonds form between parallel cellulose chains. These H bonds occur between OH groups of adjacent molecules. This creates strong crosslinks between molecules. These bonds give cellulose its strength and stability. This is crucial for its structural role. 3. Statement 3. Covalent bonds form between adjacent cellulose molecules. This is incorrect. Covalent bonds only connect glucose units within a single cellulose molecule. Adjacent cellulose molecules are held together by hydrogen bonds, not covalent bonds. The only covalent bonds are the beta 1, for glycosidic bonds within each chain. 4. Looking at the options. A. 1, 2, and 3. Incorrect. Contains false statements 1 and 3. B. 1 and 3 only. Incorrect. Both statements are false. C. 2 and 3 only. Incorrect. Statement 3 is false. D. 2 only. Correct. The correct answer is D. 2 only. Reasoning. Only statement 2 is correct. Hydrogen bonding between adjacent cellulose molecules is a key feature that * provides strength. * creates stability. * allows for structural support in plant cell walls. * enables cellulose to function effectively as a structural molecule. The other statements describe incorrect structural features. The combination of parallel chains and hydrogen bonding is what gives cellulose its structural properties. To identify alpha-glucose, we need to look for these key characteristics. 1. The OH group on the anomeric carbon, C1, should be below the ring plane. 2. The molecule should be in a pyranose, 6-membered, ring form. 3. All groups should be in the correct orientation for glucose. Looking at structure B, it has a 6-membered ring, pyranose form. The OH group on C1, anomeric carbon, is pointing downward, below the ring plane, which is characteristic of alpha-glucose. The CH2OH group is in position 6. The OH groups are in the correct orientation for glucose. The other structures either have incorrect OH orientations, have the OH group on C1 in the wrong position, have incorrect overall structures. Therefore, B represents alpha-glucose. A condensation reaction is defined as a reaction where two molecules join together. A small molecule, usually water, is removed. A new bond is formed between the molecules. Let's examine each option. A breaking of a glycosidic bond. This is hydrolysis, addition of water, not condensation. Breaks bonds rather than forms them. This is the reverse of a condensation reaction. Therefore, this cannot occur in a condensation reaction. B. Formation of a disaccharide. This is a condensation reaction. Two monosaccharides join together. Water is removed. A glycosidic bond is formed. This can occur in a condensation reaction. C. Joining together of two amino acids. This is a condensation reaction. Forms a peptide bond. Water is removed. This can occur in a condensation reaction. D. Production of a molecule of water. This is part of a condensation reaction. Water is always produced as a byproduct. This can occur in a condensation reaction. Therefore, the answer is A. Breaking of a glycosidic bond cannot occur as a result of a condensation reaction. This is because 1. Condensation reactions join molecules. 2. Breaking bonds requires hydrolysis. 3. Breaking glycosidic bonds is the opposite of condensation.
A. Consists of 4 polypeptides. This is incorrect. Quaternary structure can involve 2 or more polypeptide chains. It's not limited to exactly 4 chains. Some proteins have 2, 3, 4, or more subunits. B. Depends on the presence of metal ions. This is incorrect. While some proteins do contain metal ions are not required for quaternary structure. They may stabilize structure but aren't essential. C. Depends on the primary structure of the polypeptides. This is correct. Primary structure, amino acid sequence, determines how polypeptides fold, how they interact with each other, Asterisk the final quaternary arrangement. The amino acid sequence influences all higher levels of protein structure. D is made of alpha and beta polypeptides. This is incorrect. While some proteins do have alpha and beta subunits, like hemoglobin, not all proteins with quaternary structure have these specific types. This is too specific to be a general rule. The correct answer is C because 1. The primary structure always determines higher-order protein structure. 2. The amino acid sequence influences how subunits interact. 3. Without the correct primary structure, proper quaternary assembly cannot occur. 4. It's a fundamental principle that applies to all proteins with quaternary structure. Looking at the diagram again and analyzing why option C, molecules R and S bind to the active site of molecule P, is correct. 1. Molecule P appears to be an enzyme based on its shape. It has a larger structure. Shows what appears to be an active site, the indented region. 2. Molecules R and S are smaller molecules. Have shapes that appear complementary to different parts of P's active site. Their shapes suggest they could fit as substrates in P's active site. Both appear to be potential substrates based on their size and configuration. 3. Option C is correct because it correctly identifies that both R and S can bind to P's active site. The shapes of R and S appear to be complementary to the active site of P. This represents a typical enzyme-substrate binding scenario. The other options are incorrect because option A, Q doesn't appear to be a non-competitive inhibitor based on its shape. Option B, there's no indication of Q increasing activation energy. Option D, nothing suggests R and S are breakdown products. Therefore, C is the correct answer as it accurately describes how molecules R and S can bind to the active site of enzyme P, which is supported by their complementary shapes shown in the diagram.